Bonjour, je suis très heureuse de participer ici. But, thank you so much, Philippe. I am very, very happy. I'm from Spain, close to Barcelona, and I will talk about NMR metabolomics in this context, in the context of biomarkers and in the cardiometabolic context. So I have to start with the central dogma of molecular biology. We have been told or trained that genetics is the initial of this pathway that starts trying to flux information from the genes to the proteins that at the end are helping our cells to do their function. And the metabolites are the end of this pathway or this system. Metabolites are closer to the phenotype in the sense that are the immediate image or picture of what's happening in this information flux. So from the same genotype, we can have different phenotypes according to the exposome, what we eat, if we are smoking or not, what's our microbiome or our diet, etc. So the metabolome at the end is reflecting what's happening in this process. But we have to go farther, the central dogma, and think about a closed system in the sense that the metabolites have also actions on regulating these activate or silent genes, or activating or inhibiting the synthesis of these proteins. So at the end, all our metabolism is acting as a system. And from this point of view, the omic science appears in the sense that the information is bidirectional and it's integrated and it's dynamic and highly regulated by the metabolic context. So if we think about omics and where this word comes from, omics comes from the omni. Omni, it's the whole. So the omnivore is the animal that can eat all the things or different things. The omnipresent is the one who can be at the same time, the same in every place. So the omni, in the context of omic science, means that the whole metabolites or molecules that you can measure. And from the whole omic science, metabolomics is the youngest in the sense that it came 10 years more or less later than the genomics that start at the end of the 20th century, when the Human Genome Project finished. So once we have the technology to profile the whole of the genes or the whole of the proteins, of the whole of the metabolites, we can study the biological systems through this concept of omic science. In the case of the genes, the structure, the information, it's based on 4 molecules. So we can explore the whole genome by using 4 pieces of information. In the case of proteins, we can use 20 pieces of information, the amino acids. But in the case of metabolites, the variety or the diversity of the molecules is so high that the challenge is in how do we measure these metabolites to get a full sample set or a bit more information than the classical techniques. And here it's where metabolomics appear. In the case of approaches, there's 2 main approaches, the targeted one or the untargeted one. In the case of the targeted, it's a hypothesis-driven approach. So you are looking for some pathway or you are suspecting that these molecules are associated with a particular biological condition. And the untargeted, that you don't necessarily know what's happening there. You try to measure as many things as you can, and at the end you are generating hypotheses because you identify some associations that were not previously expected. And there's different techniques. The 2 principal techniques is the NMR metabolomics, and it's more old, and the mass spectrometry, that it's maybe the most used in the molecular biology context. Both of them are very complementary Indeed, part of our studies, we are using both, but I will discuss a bit further today on nuclear magnetic resonance in the context of cardiometabolic disease. Depending on the sensitivity that you need, you can go through NMR or you can go through mass spect, in the sense that if you need to characterize a very low concentrated metabolites, you cannot use nuclear magnetic resonance. You have to go through mass spect. But for other reasons, NMR can be very powerful. So what can offer in cardiometabolic space? I try to summarize so the main aspects, that it's very high throughput, that for a clinical point of view, that's very important. You have to be able to profile lots of metabolites for different persons in a high throughput way. It's competitive in price. It's not very expensive. If you have a project to analyze a huge number of samples, you can use NMR. It's quantitative, and that for clinical reasons, it's very relevant, because you can compare. If you have a quantity, you can, the absolute quantity, you can compare along the years, along the projects, along the studies. And that's very important for clinical applications. It's automated, fast. You can analyze any biological matrix. In fact, I would like to share that we have received a sample from bone marrow from an elephant, and that is very interesting also to know. But in the human cardiometabolic context, we are usually working by plasma or serum. We can establish population values, and that it's very important to identify if an individual is behaving very far from the normal of the population. And in particular, you can measure the advanced lipoprotein profile. And I will go a bit farther in this case for the context. We start working by NMR because of the advanced lipoprotein profile. As you know, and in the previous talk, very nice talk indeed, this slide has been more or less presented, and it shows how atherosclerosis, that it's the silent disease before the cardiovascular accident appears, that it's accumulating cholesterol in your arterial walls. And that process starts very early in our lives, and there's one moment that this plaque, the atheroma plaque, it's broke, or provoking an ictus, or it's obturing the wall, or provoking an infarct, etc. But the disease before the accident occurs is the atherosclerosis, and this process is the result of accumulation of cholesterol. That it's like a plastic, that it cannot be removed from our arteries in general. And if you have very high levels of cholesterol, this process is accelerated, as it has been explained just before. But if we take a look on the people who has already cardiovascular accidents, at least the half of the population present totally normal levels of LDL cholesterol. And that brings us a question, why? There's other factors known as residual risk factors that can explain this presence of cardiovascular accidents or accelerated atherosclerosis beyond the amount of cholesterol. And one of the factors that it's one of the main drivers of this accelerated atherosclerosis is the particles, the number of particles that can cross the arterial walls. If we take a look on the levels of cholesterol and particles of a population, a random population, we can see that there are some very concordant people that present low levels of cholesterol and low levels of particles, and people with high levels of cholesterol and high levels of particles. But there are some people who have especially small particles that are associated with increased risk. If we take a look on the amount of cardiovascular accidents accumulated across the years, we can see that the group of these particularly small particles has a higher number of accidents in the follow-up. And how are produced these smaller particles? What happens to have this pattern of smaller particles? And the triglycerides have a very important role in this process. If we think about how is an ApoB-containing particle since it is born, we have to think that our liver is constantly producing the ApoB protein. And as soon as it has enough fatty acids to be ensembled, the particle is released in the bloodstream. So if there's no fatty acids, then the protein is degraded. So it's a process that it's constantly producing very low density lipoproteins that are in our body mainly to transport energy. It's a way to get triglycerides in the rest of the body. And these triglycerides are hydrolyzed along the life, along the first day. And the rest, the 90% of the time, these ApoB-containing particles has become enriched in cholesterol and has become an LDL particle. So an ApoB, it's constituting the 10% of the time an very low density lipoproteins, and the 90% of the time, in normal conditions, it's constituting an LDL particle. But when there's some alterations in the triglycerides metabolism, this process, this clearance of the newly secreted particles to LDL, it's stopped or it's slowed. And there appears this remnant pathway. The plasma triglycerides levels are a bit elevated, and then there's an overproduction of very low density lipoproteins. There's a reduced lipolysis that is associated with hyperinsulinemia or hyperglycemia. There's an increased remnant pathway. These particles remain in our bloodstream. And there's also an increase in the protein that is transferring cholesterol esters and triglycerides among lipoprotein subclasses. So these particles, LDL particles, are interchanging cholesterol and triglycerides with the very low density lipoprotein particles or remnant particles. So they are more dense and smaller. And also the HDL, that there are more enriched in triglycerides. So all this profile, it's associated with cardiovascular disease in the sense that we have more particles, they are more time in circulation, so more prone to be oxidized. They are crossing the arterial walls, modified and aggregated in this plaque. So all these modifications together are factors that contribute beyond LDL cholesterol to the formation of the plaque. So why NMR? NMR is a technology, it's a magnet, that you can analyze lipoproteins through the lipids that are inside the particle. So the cholesterol and the triglycerides inside the particle present specific signals in the NMR spectra. And in the same way that the bells present different frequencies according to the size, the lipids inside the particles present a response that depends on the size of the particle. And that gives us an opportunity to measure from the intact plasma the number, the composition, the size of the whole set of lipoprotein profile from an intact sample. And that's why we start using NMR for cardiometabolic approach. All this information, as I mentioned, it's not one isolated parameter. It goes through a pattern. You have elevated triglycerides, but maybe some other alterations in the lipoprotein profile. And trying to summarize that in an image, we develop what we know as a lipid silhouette, that it's this representation that tries to integrate the different parameters that can be associated, including LDL cholesterol, triglycerides in LDL particles, remnant cholesterol, very low density lipoproteins, triglycerides, size of the different lipoprotein particles, and the same individual, or three different individuals with exactly the same amount of LDL particles can present totally different profile of lipoprotein alterations. So we have implemented that in clinical settings. So we have to think about NMR, that it's a technique that it's fast and cheap, that can profile traditional lipids, but also these lipoprotein advanced parameters, and also inflammatory biomarkers. I will go through in 2 slides. And the aqueous metabolome, around 12 dozens of metabolites, and also the lipidome. So it's a technology that gives you a broad image of what's happening metabolically spoken. And why are we using it, or for what? We are trying to understand some molecular mechanisms. We are using for diagnostics or to identify some biomarkers. We are using for improving the genetic screening and also for monitoring the treatment. And I will show you some examples of these cases. In the case of understanding more the molecular mechanisms, I would like to show you part of our results of the collaboration with the Hospital Sant Joan in Reus, that is a clinical hospital, that they are incorporated the technology into the clinical practices. So that includes 1,000 patients from the routine that have been analyzed by this technology. And we have stratified the population, these 1,000 individuals visiting the lipid unit, so with alterations in lipids in general, or familiar with alterations in lipids. And we have stratified according to quartiles of triglycerides. And we can see that the first quartile only have a bit of LDL cholesterol, but according to the increase of the triglycerides, we start identifying alterations in much more other parameters, in the size of the LDL particles, in the composition of the HDL particles, in the concentration of HDL particles. So the triglycerides are triggering these alterations. And that's, we can see that an individual with exactly the same amount of LDL cholesterol, that this amount of triglycerides is generating much more alterations than in the other case. It's not the LDL in that case. Another way to measure biomarkers associated with cardiovascular disease is measuring the inflammatory biomarkers. The NMR has a specific signal that allows us to measure glycoproteins that are associated with galactosamine, glucosamine, and sialic acid. And we can explore these inflammatory biomarkers that are generated in the liver, as a process responding to acute and chronic inflammatory disorders. They are activated according to the complement activation, chemotaxis, or even as a response, regulation and tissue repair. And they are increased during inflammatory process. And the main proteins that are elevated during inflammatory stages are 5 specific proteins, plasmatic proteins, that all of them are increased in inflammatory processes. So we can also profile this lipoprotein and glycoprotein together from the same sample. There's also something that I mentioned that was very relevant, that we are able to obtain population values. As in the image I have shown, I have used one of the samples that almost got a baby from 9 months, that it has 1,000 milligrams per deciliter, so it's very, very high. It's almost, I was a patient from the hospital in Reus. And we can see, of course, in that case, that it's the individual with higher levels of LDL cholesterol, of course, but we can also position this individual according to the rest of the population in our database. We have already analyzed around 50,000 people. And we are trying to identify the individual in comparison to the rest of the population. And by using that, we can also try to improve the screening, the genetic screening. And for that reason, I have prepared a couple of slides describing an algorithm that we developed by using this technology, including 2 populations. One of the populations was an adult population, including 300 people, more or less, that was testing positive for the clinical score Dutch for FH, but also was tested genetically. And then we have 300 people that 2/3 of the population was positively tested, genetics, and negative tested. And we compare also, or we validate the models by using a pediatrician cohort that includes around 100 population that were pediatric. So we developed an algorithm that was based on advanced parameters by using NMR, and we developed a model to predict the genetic testing of the both populations. So we used the adult cohort to develop the model and validate it with a pediatrician cohort. And we identified some parameters that were relevant for this prediction. For example, the presence of, I will show you here, the large HDL particles or remnant cholesterol, large LDL particles. And now we are applying that in the clinics in the sense that any time that there's some patient that is closely scoring high for FH, we are analyzing that and we are providing a score, genetic score for HF. And it's interesting to see that there's the clinical parameters are quite similar in two patients, that 350, 382 total cholesterol, LDL similar, HDL also similar. But by using this test, we can see that in one of them, there's very high levels of remnant cholesterol and not in the other. And so the probability was different of developing FH. Or also we can use it for monitoring. That I think that it's very, very important. Compared to other omics, this possibility of compare longitudinal data, it's very, very scarce, in the sense that NMR, it presents a very low variability in the experimental set. The sample is analyzed almost intact, so this variability, it's diminished compared to other omics. And that makes that you can compare the sample analyzed today with the sample that will be in 2 years, 3 years. And now it's also a patient from the hospital. It's a girl who has lysosomal acid lipase deficiency, and in that case was diagnosed at 15 years old. And we test her at 2018, 17, and 22, and we can monitor the treatment. So at the beginning without treatment, and after the years, she has improved the lipid profile. Of course, this drug is contributing to the reduction of cardiovascular disease, and also we can apply NMR-based metabolomics to the longitudinal processes. We can also do it in clinical trials. In clinical trials, we are collaborating with other pharma companies that are trying different drugs. In that case, it was a drug for NASH, and that was the placebo group, and different doses in 1 milligram saroglitazar, 2 and 4. And we can see that the basal is in orange, and the post-intervention is in green, how the dose is responding, or how the lipoprotein profiling is responding to the dose in that case. And finally, I want to present a rarity. I would like to show you the sample of the oldest woman in the world when we analyzed her blood. That was 116. Here the picture, it's 115. But she was very, very curious in terms of metabolomics. We analyzed the lipoproteins, the glycoproteins, also the aqueous metabolome, and also some lipidomics of this woman. And we compare to the group more similar to her, that was all ladies, but only beyond 8 years, so a bit different in age in the sense that from 115. But we can see that in her case, that it's in orange, the values were, for example, in the case of LDL cholesterol, was almost comparable to the ladies over 80. But when we take a look on her LDL particles, we could see that she had no small LDL particles. So maybe that's the reason that she was protected against cardiovascular disease. We don't know. In that case, I would like to remind you that the metabolomics goes beyond genetics and proteomics. It's providing real-time insights into the biological process. Metabolomics plays a dynamic role in regulating gene expression and protein activity, that it's changing the dogma, the central dogma. NMR metabolomics offers a unique advantages, also disadvantages, but in the case of advantages, it's high throughput, quantitative, scalable, and include advanced lipoprotein profiling that is relevant for cardiometabolic disease. We can use it for improving genetic screening, enable longitudinal monitoring, and supports population stratification. At the end, I think that it's part of these tools that will be part of a precision medicine, that it's transforming diagnostics by integrating metabolomics together with genetics, proteomics, and clinical data. So thank you for your attention, and merci beaucoup.
Thank you, Nuria, for this fascinating talk. Are there any questions in the audience? Nuria, maybe you can just say if there are some questions. 5 seconds, don't escape yet. No, I will start maybe. I didn't see the pattern of the different lipoprotein that measure, if you are able to measure Lp(a).
The problem with Lp(a) is that NMR, it's looking at the molecules that are in the liquid state. So we can see the lipoproteins through the core, through the melted cholesterol and triglycerides inside. And the Lp(a), the difference is in the protein. So we cannot distinguish if the cholesterol is transported by Lp(a) or not. And on the other side, we have to think that the total amount of particles, around 10% is from Lp(a). So in the cases of the higher number of Lp(a). So from an individual who has high levels of cholesterol, the LDL particles are around 2,000. And the number of Lp(a) are around 100. Then it's very difficult to distinguish. The amount of cholesterol transported by these particles is 10%, less than the 10%. So in several clinical trials, we have asked to measure Lp(a), and we have put efforts to do it, but we are using ELISA at the end, combined with the other, and thank you.
Okay. Other questions? Okay, thank you, Nuria. Ah, et une autre question.
Thank you for this interesting talk. I have a question that maybe you already replied, but I was wondering, especially during diabetes, we can have also other post-transcriptional modifications of apolipoproteins, such as glycation. Would you be able to detect this kind of modification?
We are not monitoring glycation. We are monitoring glycosylation that is specific. So in this peak that it's from the glycosylated proteins, we only can profile the glycosylation. The exposure to the sugar that may drive to glycation, we can measure the sugar. We can predict glycated hemoglobin, but I think that it's not the best technique to do it. We can combine it with others, but it's a kind of that are not visible by NMR. Thank you.
Okay, thank you very much. Thank you.